Welcome back to the Deep Dive. We're here again to take some pretty complex science and boil it down for you.
Yeah, make it high-yield knowledge you can actually use.
And today we're diving into something really elegant, I think. It's how bacteria, like E. coli, manage their genetic resources, their budget, basically.
It really is like an engineering marvel when you look closely. I mean, think about just one E. coli cell. It has the blueprints for maybe 4,000 different polypeptides.
Wow, 4,000.
Right. But here's the kicker. The amount needed varies wildly. Some proteins, maybe the cell only needs 5 copies. Others, like ribosomal proteins, it might need 100,000 copies.
Huge difference.
Massive. So the big question we're tackling today is, how does this tiny organism figure out, just based on what's floating around it, how to efficiently turn that production dial way up or way down?
Okay, so our mission for this deep dive is to walk you through the core ideas of bacterial gene regulation. We'll start with the historical biggies, the lacanthrep operon.
The ones everyone learns first, yeah.
Exactly. And then we'll get into some of the more modern discoveries, stuff about how RNA itself can actually regulate genes.
But before we dive in, we really need to nail down 4 key ideas. These terms sort of define how the whole system is put together. If you get these, the rest makes a lot more sense.
Okay, lay them out. What's the first way we categorize these systems?
All right, first is how the system responds to the environment. So a system can be inducible. That means it's normally off, silent, but it gets turned on when, say, a specific food source shows up.
Like lactose. If lactose appears, you need the enzymes to break it down.
Precisely. You induce the system. The flip side is repressible. The system is normally on, actively making something, maybe an amino acid.
Right, something the cell needs to build.
Yeah, but if that amino acid suddenly becomes plentiful in the environment, the cell needs to stop making it. So the system gets turned off or repressed. That saves energy.
Okay, inducible or repressible, that's based on response. What's the second pair of terms?
The second pair describes how the switch actually works. So you have negative control. This means there's a regulatory molecule, usually a protein, that literally binds to the DNA and shuts off transcription. It's like a roadblock.
Okay, negative means it stops things.
Yep. And then there's positive control. Here, a regulatory molecule binds to the DNA and actively stimulates transcription. It helps RNA polymerase get started or work faster. It's like an accelerator.
Got it. So inducible repressible tells you when it happens, and negative positive tells you how the regulator molecule acts.
Exactly. And pretty much every bacterial regulation system is some combination of these 4. It's all about adapting quickly and efficiently.
All right, let's put this framework to use. Where should we start? The classic, right? The lac operon.
Absolutely. Jacob and Monod's work. It's the textbook example of an inducible system under negative control. It's all about handling lactose.
So walk us through the parts. What are the key components here?
Okay, you've got the genes that actually do the structural genes. There are 3. lacZ, which makes an enzyme called beta-galactosidase. That's the one that cuts lactose into glucose and galactose.
It's the main job.
The main job. Then lacZ, that makes permase, which is like a gatekeeper. It helps get lactose into the cell. And lacO, which makes transacetylase, its role is a bit less critical for basic understanding, but it's part of the set.
And the crucial thing is they're all lined up together.
Yes, and they're all transcribed together onto one single long piece of mRNA. We call that a polycistronic mRNA. This is super efficient because one on switch controls all 3 enzymes needed for the pathway.
Coordinate regulation. Makes sense.
Yeah.
So what controls that on switch?
Right next door on the DNA, you have the regulatory sequences. First, there's the lacI gene. This isn't part of the operon proper, but it makes the key regulatory protein, the repressor.
Okay, the negative controller.
Exactly. Then you have the promoter, the P site. That's where RNA polymerase wants to bind to start transcription. And right next to that, overlapping it slightly, is the operator, the O site.
And this is where we need that cis-trans distinction you mentioned.
Perfect time for it. The operator O and the promoter P are specific locations on the DNA. They can only affect the genes located right beside them on that same DNA molecule. So we call them cis-acting sites.
They have to be in cis nearby.
Right. But the repressor protein, made by the lacI gene, that's a molecule that floats around in the cell. It can travel and bind to any appropriate operator site, even on a different piece of DNA, like a plasmid. We call that a transacting
Okay,
factor.
cis sites, trans factors, got it. So let's put it together.
Yeah.
Negative control. What happens when there's no lactose around?
All right, no lactose. The lacI gene is always quietly churning out its repressor protein. This repressor protein, in its default shape, has a high affinity for that operator O sequence on the DNA.
So it binds there.
It binds there. tightly. And because the operator physically overlaps the promoter where RNA polymerase needs to bind, well, the repressor acts as a physical roadblock. RNA polymerase just can't get on.
Blocked. So no transcription of lacZ, Y, or at all.
Well, exactly. The system is off. Negative control in action. And it's inducible because it's normal off.
Okay, now let's introduce lactose. The cell takes it in using the few permease molecules that are always around. What happens then?
Some of that lactose gets converted inside the cell into a slightly different form called allolactose. And this molecule is the true inducer.
Not lactose itself, but its isomer.
Correct. Allolactose finds the repressor protein, which is maybe bound to the operator, maybe floating around, and it binds to a second site on the repressor.
The allosteric site.
Precisely. Remember we said the repressor is allosteric? Binding allolactose causes the repressor to change its 3D shape. Critically, this new shape has very low affinity for the operator DNA sequence.
So it falls off the DNA.
Yeah.
Or can't bind in the first place.
Both. If it was bound, it lets go. If it wasn't bound, it can't grab on now. The operator site becomes clear.
And that means?
RNA polymerase can now bind to the promoter and start transcribing the lacZ, YO, and $ genes into that polycistronic mRNA. The enzymes get made, and lactose metabolism begins. The presence of lactose induced the system.
That's really elegant. But how did Jacob and Monod prove this model? It sounds good, but...
The beauty of bacterial genetics. Mutations were the key. They isolated mutants with weird lactose metabolism and figured out what went wrong.
Okay, give us some examples.
Sure. Take a lacA mutant. The minus means the repressor protein made by this gene is broken. It can't bind the operator at all.
So even without lactose?
The operator is always open. RNA polymerase can always bind. The genes are transcribed all the time, whether lactose is there or not. We call that constitutive expression. Always on.
Okay, that shows the repressor's job is to turn things off. What about the operator site?
Good question. They found lacA mutants, C's for constitutive. Here, the DNA sequence of the operator itself is mutated.
So the binding site is messed up.
Exactly. Even a perfectly normal repressor protein made by a functional lacA gene simply cannot recognize or bind to this altered ulticellular sequence.
Result, constitutive expression again.
Yeah.
Always on, because the off-switch binding site is broken.
You got it. And one more really informative one, lacZ. The S stands for superrepressor.
Superrepressor, what does that do?
This mutation changes the repressor protein in a different way. It can still bind the operator perfectly fine, but its allosteric site is broken. It cannot bind the inducer, allolactose.
Oh, so even lactose is present.
The repressor stays stuck on the operator. It never gets a signal to leave. The genes remain permanently off, permanently repressed, even with tons of lactose around.
Wow. Okay, those three mutants together really map out the functions of I, the repressor protein, and O, the DNA binding site.
Absolutely. They were critical proofs for the negative control model.
But wait, there's more to the lacOperon story, right?
Sure.
You mentioned positive control earlier.
Yes. Because even with the repressor gone, transcription isn't automatically super high. The cell has a preferred sugar, glucose. It's easier to metabolize.
So it doesn't want to bother with lactose if glucose is available.
Pretty much. This phenomenon is called catabolite repression. Basically, glucose represses the activation of operons for other sugars, like lactose. The cell needs another layer of control. Turn the lacOperon on fully only if lactose is present and glucose is absent.
Okay, so how does it sense glucose levels? And how does that link to turning the lacOperon up? That sounds like positive control.
It is. It involves a protein called Catabolite-Activating Protein. By itself, CIP doesn't do much. But when glucose levels are low, another molecule builds up in the cell. Cyclic AMP, or CAMP.
CAMP, I've heard of that as a signaling molecule.
It is. And here, CAMP binds to CAP. This CAMP-TAP-K complex is the active form. It then binds to a specific DNA site near the lac promoter.
Okay, so CIP binds DNA, but only when activated by CAMP. What does binding there do?
It acts like a helper for RNA polymerase. The lac promoter is actually intrinsically kind of weak. RNA polymerase doesn't bind it very efficiently on its own. But when the CAMP-CAP complex is sitting there, it interacts with RNA polymerase and basically gives it a big boost, helping it bind much more tightly and initiate transcription much more frequently.
Ah, so it stimulates transcription. That's positive control.
Exactly. High levels of transcription require both the repressor to be gone, lactose present, and the Cy-K-CAM complex to be bound, glucose absent.
And what happens if glucose is present?
When glucose is abundant, it triggers a pathway that inhibits the enzyme making CAMP-CAM adenyl cyclase, so Cy-CAMP levels plummet.
No sync-P means no active CAMP-P complex.
Right. Even if lactose is present and the repressor is off the operator, without that cyclic-CAMP boost, transcription from the lac promoter is very low, minimal. The cell focuses on the preferred glucose.
Okay, that makes perfect sense. It's like a two-factor authentication system. Lactose present and glucose absent for the green light.
That's a great analogy. And you know what's amazing? Decades later, X-ray crystallography lets scientists actually see how the repressor works at the molecular level.
Oh yeah. What did they find? Did it match the genetic model?
It confirmed it beautifully and added incredible detail. The functional repressor isn't just one protein. It's actually a homotrimer, 4 identical subunits working together.
4 of them, okay.
And it doesn't just bind to that main operator we talked about.
Yeah.
It turns out there are 2 other weaker operator sites nearby, O22 and O32. The tetramer binds to the main one and one of the auxiliary ones simultaneously.
Wait, it binds 2 spots at once. How does that work if they're separated on the DNA?
It forces the DNA in between about 93 base pairs to jut out, forming a repression loop.
It loops the DNA.
Yes, it literally bends the DNA into a loop structure. This loop physically blocks the promoter region much more effectively than just sitting on one spot. It makes it almost impossible for RNA polymerase to get access.
That's incredible. A physical loop of DNA as the off mechanism.
Molecular mechanics at its finest. Really stunning structural confirmation of the genetic predictions.
Okay, so that's the lac operon. Inducible, negative control via the repressor loop, and positive control via KPCMP. What about the other classic, the TREP operon?
Right, the TREP operon. This is our model for synthesis, or enablism specifically, making the amino acid tryptophan. And it's a classic example of a repressible system under negative control.
So contrast it with lac right away. Repressible means it's normally.
Normally on. Tryptophan is essential. The cell usually needs to make it. So the default state is transcription is happening.
Why is it on by default? Is the repressor different?
Yes. The TREP repressor, made by the TRPR gene, is synthesized in an inactive form. By itself, it cannot bind to the TREP operator site.
Ah, so the roadblock isn't there initially.
Correct. RNA polymerase can freely bind the promoter and transcribe the 5 structural genes needed to synthesize tryptophan, EDCBA. Again, it's a polycistronic mRNA.
Okay, but it's repressible, so when does it turn off?
It turns off when tryptophan itself becomes abundant. Maybe the cell finds tryptophan in its environment or it's made enough.
So tryptophan is the signal. What does it do?
Tryptophan acts as a corepressor. It binds to that initially inactive repressor protein.
Another allosteric interaction.
Exactly. Binding tryptophan causes the repressor to change shape into an active conformation. And this active repressor tryptophan complex can now bind tightly to the TRP operator.
And block RNA polymerase, just like the lac repressor did.
Precisely. It blocks transcription. So when tryptophan is plentiful, the cell stops wasting energy making more. Tryptophan itself triggers the shutdown. Notice the difference. Allolactose, from lactose, inactivates the lac repressor to turn the system on, while tryptophan activates the TRP repressor to turn the system O off F.
Okay, clear contrast. Inducible versus repressible. Allolactose as inducer versus tryptophan as co-repressor.
You got it. But there's another layer of control on the TRP operon, something even more subtle and rapid.
Don't tell me. More complexity.
Well, yes, but it's fascinating. It's called attenuation.
Attenuation. Okay, what does that mean?
It means that sometimes transcription starts, but it gets stopped prematurely very early on. It's like weakening or attenuating the signal before it even gets to the main genes.
Stopped after it starts. Where does this happen?
It happens within the leader sequence of the TRP mRNA, about 140 nucleotides in, well before the actual structural genes.
Leader sequence.
Yeah.
Like before the coding part.
Exactly. And this leader sequence isn't just spacer DNA. It contains some really important features, including 2 codons right next to each other that code for tryptophan.
2 tryptophan codons.
Yeah.
And the leader sequence, why?
That's the sensor. The cell uses the act of translating this leader sequence to figure out how much tryptophan is actually available right now in the form of charged tRNA molecules. tRNA, CRIP.
Whoa, okay. So the ribosome is involved in regulating transcription. How?
It's a race, essentially. The leader sequence RNA has regions that can fold and base pair with each other to form hairpin loop structures. There are 4 key regions, let's call them 1, 2, 3, and 4. They can form different pairs, 1, 2, 2, 3, or 3, 4.
Okay, different possible solds.
And crucially, the 2-3 pairing and the 3-4 pairing are mutually exclusive. If 2 pairs with 3, then 3 cannot pair with 4. And the 3-4 hairpin acts as a terminator signal for transcription, like the ones we see at the end of genes. The 2-3 hairpin, however, is called the anti-terminator.
Terminator versus anti-terminator.
Got it.
How does the ribosome and tryptophan level influence which one forms?
Okay, scenario 1. Tryptophan is abundant. That means there's plenty of charged tRNA TRPA. When the ribosome starts translating the leader sequence, it reaches those two UGDI-codons.
And it finds the tRNAs easily and zips right through them.
Exactly. It moves quickly past the UGDI-codons and covers up region 2 of the mRNA. Because region 2 is covered, it can't pair with region 3. This leaves region 3 free to pair with region 4.
Forming the 3,4 terminator hairpin.
Bingo. The terminator forms, signaling RNA polymerase, which is just a little bit ahead, to stop transcription. Attenuation occurs. The structural genes aren't transcribed, even though the repressor might not even be bound yet.
Wow. A quick-acting secondary off switch if tryptophan is high. Now, what if tryptophan is scarce?
Okay, low tryptophan means low levels of charged tRNA OA. The ribosome starts translating the leader, hits those two UGDI codons.
And it stalls. It has to wait for a rare charged tRNA.
Precisely. It pauses, sitting right there over region 1. Because it's stalled before covering region 2 is now free. And what does it do?
It pairs with region 3, forming the 2,3 anti-terminator hairpin.
You got it. And because 2 is paired with 3, region 3 cannot pair with region 4. The terminator hairpin doesn't form.
So the stop signal is prevented.
Right. RNA polymerase just keeps chugging along, transcribing right through the leader sequence and into the structural genes E, D, C, B, A, D. The cell makes the enzymes it needs to synthesize more tryptophan.
That is incredibly clever. Using the speed of translation of specific codons in the leader sequence to control the structure of the mRNA and decide whether to continue transcription.
It's a beautiful example of kinetic control and RNA structure playing a direct regulatory role. It provides a much more sensitive, graded response to tryptophan levels than just the repressor alone.
Okay, so we've got repressors, activators, copressors, inducers, loops, attenuation. Is that it?
Not quite. The discoveries continued, revealing even more ways RNA itself controls gene expression, often without any proteins involved at all. We need to talk about riboswitches.
Riboswitches. Okay, the name suggests RNA acting as a switch. How do they work?
They're typically short sequences found in the 5-foot untranslated region, 5-foot UTR, of an mRNA molecule, kind of like the leader sequence we just discussed.
So part of the mRNA itself.
Yes, and the key is this RNA sequence can directly bind to small molecules, things like metabolites, vitamins, ions, no protein needed for the binding.
The RNA binds the metabolite directly.
Directly. The binding region is called the aptamer. When the specific small molecule ligand binds to the aptamer, it causes the RNA structure to change its folding pattern.
Another conformational change, but this time in the RNA itself.
Exactly. And this change affects the second part of the riboswitch, called the expression platform. Often, ligand binding causes the expression platform to snap into a terminator hairpin structure, just like in attenuation.
So if the metabolite is present, it binds the RNA, forces the terminator structure, and shuts down transcription of the downstream gene.
That's a common mechanism, yes. It switches the RNA from an anti-terminator form to a terminator form. But riboswitches can also work at the level of translation.
Oh.
Binding the ligand might cause the RNA to fold in a way that hides the ribosome-binding site, RBS. If the ribosome can't bind, translation can't start, even if the mRNA was fully transcribed.
So RNA sensing a metabolite and then either stopping its own synthesis or stopping itself from being translated. That's really direct feedback.
Incredibly direct. It's a widespread mechanism in bacteria for sensing nutrient availability and adjusting gene expression on the fly.
Okay, one last category you mentioned earlier, RNA regulators that aren't part of the mRNA they control.
Yes, the small non-coding RNAs, or sRNAs. These are short RNA molecules, maybe 50 to 500 nucleotides long, that are transcribed from their own genes.
So they're separate molecules, not part of an mRNA UTR.
Correct. And they work by base pairing with target mRNA molecules, usually somewhere near the 5-foot end.
They bind to the mRNA. What does that achieve?
They can act as either negative or positive regulators, mostly at the level of translation.
How do they act negatively?
Often, an sRNA will bind to the region of the target mRNA that includes the ribosome binding site, RBS. By physically covering up the RBS, it prevents the ribosome from initiating translation.
So it blocks protein synthesis from that mRNA.
Exactly, negative regulation, but they can also be positive regulators.
How does that work?
Sometimes an mRNA molecule might naturally fold up on itself in a way that hides its own RBS, preventing translation. An sRNA can come along, bind to a different part of that mRNA, and break up that inhibitory secondary structure.
Oh, so the sRNA binding unmasks the RBS.
Precisely. By changing the mRNA structure, it makes the RBS accessible to the ribosome, thereby enhancing translation. Positive regulation.
Can you give an example of an sRNA in action?
Sure, a well-studied one in E. coli is called rIHB. This sRNA is produced when iron levels in the cell are very low.
Okay, low iron triggers RIHB. What does RIHB do?
Iron is used in lots of enzymes. When it's scarce, the cell needs to prioritize. rIHB acts as a negative regulator. It binds to the mRNAs of several enzymes that use iron but are considered non-essential when iron is limiting.
So it blocks the translation of less important iron-using proteins.
Yes. By binding their mRNAs and blocking the RBS, it shuts down their synthesis, effectively conserving the precious little iron available for the most critical cellular functions. It's resource management via RNA interference.
Wow. It's another layer of sophisticated control. It seems like bacteria have evolved this incredibly diverse toolkit for gene regulation.
Absolutely. From proteins binding DNA to DNA looping to RNA sensing metabolites to RNA controlling RNA, it's multi-layered and highly adaptive.
Okay, let's try to quickly recap the main points we've hit today.
Sounds good. We saw that bacterial gene regulation is fundamentally about efficiency and adapting to the environment.
Right. We started with the lac operon as the classic model. It showed us inducible systems, negative control through the repressor protein binding the operator.
And positive control through this CKP complex responding to glucose levels, enhancing transcription only when needed.
Then we contrasted that with the trap operon, our model for a repressible system, where the end product, tryptophan, acts as a co-repressor to activate the repressor and turn the system off.
And we saw the amazing fine-tuning mechanism of attenuation in the trap operon, where the ribosome's speed across leader codons determines whether an mRNA terminator hairpin forms, stopping transcription early.
And finally, we touched on the purely RNA-based regulation. Riboswitches, where mRNA directly binds metabolites to change its own structure and control expression.
And small non-coding RNAs, sRNAs, which act as transacting regulators, binding to target mRNAs to either block or enhance translation.
It's just remarkable how these physical structures, proteins, DNA, RNA, interact with such precision. Thinking about that repression loop in the lac operon, where the DNA itself is bent by the repressor.
Or those alternative hairpin structures in the trap leader mRNA, snapping into terminator or anti-terminator forms based on translation speed.
Yeah. What does that intricate molecular choreography, these physical shapes and changes, tell you about, I guess, the fundamental mechanics of life at this level?
To me, it reveals an incredible elegance. The structure is the information processor, the shape of the molecule, how it changes when it binds something else that is the computation. It integrates environmental signals, metabolic status, resource availability, all through these dynamic physical interactions. It's biology leveraging physics and chemistry in the most efficient way imaginable.
The structure is the logic gate, a fantastic way to put it, an amazing deep dive into bacterial control systems.
It was fun, always fascinating to revisit these foundational mechanisms.
Absolutely. Well, that's all the time we have for today. Thank you for being a part of our little last-minute lecture family.